Hi, welcome to another edition of whatever this is. Today's concept is going to be about the mutations in DNA. So far we've learned about the DNA basic structure, how and why it packages itself into the nucleus. That's a concept called epigenetics. We've learned about gene expression, which is when a gene gets expressed and turned into a protein through the process of transcription and translation. And today we'll round out this chapter with mutations. So let's get started. All right, so a mutation is a random change in the order of nucleotide bases in DNA. That random is a very key word. We can genetically modify things in a lab, which is the case of the seedless watermelon, but for our purposes, we're going to think of mutations as a very random change to the sequence of those bases, those A's, those T's, those C's, and those G's. It can result from any number of mutagens, and a mutagen is anything that causes a mutation to occur. So for example, you have ionizing radiation, asbestos, radon, cigarette smoke. Radon is something that occurs in groundwater, so that's kind of important here in New England. It comes up through the rocks, the uranium in the rocks, and it degrades into radon, which then gets emitted in shower steam or into your drinking water, which can be bad. So a mutagen is anything that causes a mutation. So one point where mutations are likely to occur is during DNA replication, which is what this animation is trying to show. So in this case, you have a DNA double helix coming in. This enzyme here called helicase is going to spin so fast that it actually separates the two strands. You can see one going this way off to the right, and then another one going straight ahead like that. Now DNA can only be, when you have a single strand and you're trying to find complementary strands, it can only be done so in one direction, which is why this one gets done nice and quickly. So you have a small single strand here, but it only gets done in one direction. And this one coming out the back end is a long strand of exposed bases, and the problem here is that those exposed bases can be mutated. This is where a mutation is likely to occur. I think if someone's coming to my head. Okay, so that's a problem. That's where DNA replication can be a problem. So there's three types or three methods of mutations that we're going to learn about. They're referred to as SNPs, or single nucleotide polymorphisms. This is when one single base is going to be disturbed. So the first possible way to do this is something called an insertion. This is when an extra base is added to the sequence. So here's our original strand, CAT, 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 CAT. This would code for histidine, 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 all the way down this amino acid sequence. In our mutated sequence, this C has been, or between the T and the A, between T and the C, an A has been inserted. And so what's going to happen is this is going to throw off all the codons from this point forward. Instead of being CAT here, it's ACA, because the C got shifted to the right one, then the A got shifted to the right one, and everything after this got shifted right by one position. So that's going to code for a different amino acid here, and then this point going forward, it will be the different sequence of amino acids. This is extremely problematic. This will result in a totally either non-functioning protein at best case, or in the worst case, that protein is going to be very detrimental to the health of the cell. Similar to an insertion would be a deletion. So in a deletion, a base is removed from the sequence. So here we have our same original sequence, histidine, 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 but this A right here between the C and the T gets deleted. And as a result, the T shifts to the left, then the C shifts to the left, and the A shifts to the left, and everything after that point of the mutation is different from what it should be. The CTC would code for leucine, and then ATC would be isoleucine, 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 instead of histidine, which is what it should be. So a deletion can also shift the entire process of protein synthesis off by one nucleotide, resulting in new codons. This concept of shifting everything down by one to the left or to the right is known as a frame shift. If you think of each codon as a single frame in the whole sequence, when you shift one of the nucleotides out or in, that's going to change the frame. So we call it a frame shift. It's a very, very detrimental, very bad and negative mutation. Our third method of mutation is called a substitution. Now the good thing about a frame shift caused by an insertion or deletion is that mutation will be picked up pretty quickly by the cell and hopefully dealt with. They can go through and cause problems, but sometimes they get picked up. A substitution, on the other hand, is much trickier. A substitution is a very small mutation, and so what will happen is it will get, it's possible that it will get passed through our proofreading system, and it will stay a part of the mutation. So there's three types of mutations that can occur from a substitution. The first is a silent mutation, the second is a missense mutation, and the third is a nonsense mutation. Let's take a look at what those three things are. A missense mutation is the most common type of mutation to occur. So here is our original sequence. The DNA template strand would be this, mRNA would be transcribed into this, and then this would be translated into glutamine right here in our second to last codon. This amino acid sequence would equal a protein that would give our red blood cells this nice shape, almost like a donut without a hole in the middle. This has a lot of surface area on which it can carry oxygen to your cells and to wherever oxygen needs to go. In a mutated sequence, the CTT, if this T gets swapped out for A or anything happens to this codon, it's going to transcribe to GUA, which will translate to valine. Now this mutation is bad because it will still produce a protein, but that protein will result in a different shaped red blood cell. Instead of that nice round oxygen carrying blood cell, you have what's called a sickle cell. This can't carry nearly as much oxygen, and also it gets hung up on your capillaries as it travels through your blood vessels. So that can be pretty bad. A nonsense mutation is also very, very disruptive. It basically doesn't allow protein to be created. So here we have a section of a protein, some amino acid sequence where we have glycine, 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 all the way down. In this case, this substitution substitutes a C right here for a T, and that TAG will code for a stop amino acid. It's a stop codon. So what would this instead of having a nice fully fleshed out protein, it gets stopped prematurely, and the protein becomes probably non-functional or maybe partially functional, but it would be bad. A silent mutation, on the other hand, is not that big of a deal. So here we've got a couple of codons. The last one is CAA, which transcribes to valine. If an A gets swapped out for a thymine, adenine to thymine, then the CAT will still code for valine, and so it's not that big of a deal because you get the same amino acid sequence, and which means that you have the same protein as well. Alright, so here's a summary of those mutations. These insertions and deletions would lead to a frameshift mutation. The substitutions can lead to silent, nonsense, and missense mutations. I'll print this out for you guys and you can glue it into your notebook. Alright, I want to take a time out for the bigger picture here. The whole point of the life science unit is to show you how evolution works. That's our last topic in this unit. But the very foundation of evolution is genetic variety or genetic diversity. It's this variety that gives a species its set of genetic tools that they can use to overcome threats of extinction. The more diversity a species has, the more able it is to adapt. If you have a lot of genetic diversity, that means that you might be able to adapt to environmental pressures because some members of your species might be able to handle those changes better. There are 4 ways that biology encourages genetic diversity. The first one we're going to learn about is mutations. So remember this slide, it's a major concept in the life science unit. We're going to learn about the other 3 in the upcoming chapters. Alright, so lastly, if you have a mutation, there's a couple things that could happen. You could have an improvement to the organism as we saw in our sentence right here. So in our substitution and missense mutation, we have an improvement to the actual function of the sentence. So every now and then a mutation will come along and improve a protein, which will help an animal survive better. Most likely you'll have a harmful change. This is called a deleterious mutation. An example of that would be the sickle cell disease, where it still functions, but it's not that good. It's not as good as the original. Or it could be that the problem is very serious and it would cause or lead to a loss of life. The third outcome is that there's no meaningful change whatsoever. That would be the case of a silent mutation as shown down here. GGA and GGT still code for the same amino acid protein. Okay, so that's all for mutations. Thank you very much for listening.